All right, here we go. This is host defense part 2, still looking at innate immunity. We're going to be toggling back and forth between this screen and a couple of others. This seems to be the best format to do this recording. And let me write some things down and draw some images for you. So bear with me for some of the transitions. We will be covering in this set of slides the rest of chapter 12. This is the second line of defense. In lecture on Thursday, we talked through the different cells of the immune system, and today we'll see how they function in the inflammatory response. We'll also have a brief look at the interferon system and the complement system, but we won't spend a lot of time on there. There's a key concept for this set of slides, and this is the events of inflammation, including phagocytic action. We will write this out and draw this out, and I'll do the best that I can to help explain the measly pictures in your textbook. So before we get into the second deck of slides, remember that we had a few slides from that first deck that we wanted to try to finish talking about here. So I've already cut and added them right here to this set of slides for this PowerPoint presentation. So again, we ended on looking at all of the cells in the second line of defense. Well, actually all of the white blood cells we have. So now let's talk about what they do and how they communicate with one another. So every cell in our body communicates through cell surface receptors that bind to particular ligands or ligands, depending on tomato. Ligands can be any type of molecule depending upon what that receptor is finely tuned to bind to. This binding, just so you know, is like that specificity we talked about with enzyme substrate complexes. In this case, the enzyme is a little bit like a surface cell receptor, and then that substrate is like that ligand, just to get that concept in your mind. And for cell-to-cell communication, these receptors function actually differently than enzymes. Once they are bound, the binding of receptors on the surface of the cell will elicit some type of cellular response. Oftentimes, that signal can be propagated all the way down to the nucleus to change the gene expression of that cell. Sometimes it interacts with proteins that are in the cytoplasm of that cell and changes the function or physiology. So we're going to look at some examples of this in how our white blood cells and how some of our epithelial cells sense and respond to bacterial markers. And we call these markers pathogen-associated molecular patterns. Right, so pathogen-associated molecular patterns, or PAMPs, and we're going to write down some more vocabulary terms. So again, bear with me, we're going to sort of toggle through a couple of things here. All right, so we started with abbreviations on Thursday. Let's add those abbreviations here already. So we talked about DCs as our dendritic cells. We talked about M, and I can't really do that phage symbol, so I'm just going to do M0, and I'm sorry about that, but that's our macrophages. And today we're going to add some more to it. So we just mentioned that microbes are recognized by particular molecular patterns. And so those that are found on the surface of pathogens we call PAMPs. These are on the surface of pathogens. This would be pathogen-associated molecular pattern. I'm going to put another one, the same thing, MAMP. This in immunology is actually really more what we use. This is the same concept, but it's a microbial associated molecular pattern. I don't remember which one your textbook uses, but both of them essentially mean the same thing. So the concept of a molecular pattern is a molecule that is repeated over and over and over again. Hopefully you're thinking already about some. If I were in front of you, I would be asking you, but perhaps you can come up with some on your own. A couple of great examples that our immune cells and our epithelial cells are very well adapted to respond to are the repeating patterns of lipopolysaccharides, or LPS. Remember, these are lipids, carbohydrates, and protein. Repeats over and over and over. And every bacteria has their own particular LPS, but in general, the pattern is the same. You remember that only one class of bacteria has LPS. Indeed, that's gram-negative bacteria. So we have the ability to respond to pretty much every gram-negative bacteria that comes in with that that particular molecular pattern, that LPS, and I'll explain to you how that binding works. Another example, though, the peptidoglycan, another molecular pattern. So a molecule or a molecular pattern, binding pattern that is repeated over and over and over again. In this case, we're talking about NAG and NAM sugars that are repeated over and over, right, or repeats. So there are a number of other ones that fit into this category. In fact, we have the ability to recognize about 9 different types of molecular patterns. This is considered a non-specific recognition, though. Why? Well, because it doesn't matter what the specific type of bacteria is, what the species or even genera is, the name of the genus. It matters if it's a gram-positive or gram-negative, or if it's bacterial versus viral, but that's a very broad classification. So this is still part of our non-specific response unit. These molecular patterns are recognized, and they're recognized by particular receptors we call PRRs, or pattern, like these I just talked about, recognition receptors. There are a number of different types of pattern recognition receptors that our cells have on the surface of them. One class in particular that we will talk about here is TLRs. I know a lot of abbreviations, but this is what immunologists love to do. There's a lot of different vocabulary or new vocabulary terms you'll be learning here. TLRs, this is toll-like receptors. All right, so what are toll-like receptors? Well, again, they're a class. These are a class of PRRs, or pattern recognition receptors, and they are on the surface, found on the surface of our epithelia, and phagocytic cells. So we talked about this on Thursday, that that whole layer of cells that separates really the outside from the inside of our body is exposed to all kinds of environmental stuff, right, including bacteria and viruses, and that normally they're protected with mucus, but if there is a breach in that physical barrier and the bacteria or viruses penetrate all the way to the surface of the epithelium, our surface epithelial cells have these receptors that are adapted to respond very, very well and really robustly to the engagement of their, we call it a cognate, or to their specific pattern that they recognize. So for a classic example of these is toll-like receptor number 4 that binds with high degree of specificity to lipopolysaccharide. So a great example, you don't have to remember it, but it's a great example, is toll-like receptor class number 4. And this binds, and I'm going to use this colon to indicate the binding of receptor to ligand. So TLR4 binds very well to lipopolysaccharide, and this binding will then elicit a response. So let's go back to our slides here, and we'll take a look at what we are talking about with this binding eliciting a response. Okay, whoops. So here we are again with our specific pathogen-associated molecular pattern. And I'm going to see if I can get... Okay, again, so these toll-like receptors bind to these pathogen-associated molecular patterns, or the toll-like receptors, right? And they're found on the surface of our white blood cells, like macrophages and other phagocytic cells here, but also on the surface of our epithelium. And again, once bound, the receptors transmit a signal to the nucleus, which includes altering the gene expression here for certain cell-to-cell communication proteins and the release of very particular types of proteins. We'll look at that in just a minute, but let me show you an image of what we're talking about. So again, let's use this guy here. So when, for example, our toll-like receptor number 4, like this guy in the image here, detects its particular molecular pattern that's on that bacterial cell. For example, again, lipopolysaccharide would be the ligand for toll-like receptor number 4. This binding, and it must be a particular interaction, this binding will make this receptor begin a signal that's propagated inside that cell. For these pattern recognition receptors, almost always they will change that cell to produce something called pro-inflammatory cytokines. pro-inflammatory cytokines, or cytokines, particular molecules that induce further host defense response. So here we go, production of specific proteins that alert other components of host defenses. In this image, you can see a few of the other toll-like receptors that we have, or pattern recognition receptors that are able to bind to particular bacterial or viral patterns, molecular patterns. Another, we talked about peptidoglycan as another molecular pattern. The flagellin protein, so that filamentous component of the flagellum has flagellin protein in there, and that is a pattern that is recognized by our surface cells and our white blood cells, and other things like nucleotide sequences. And there are a few others as well. But the same thing happens every time a toll-like receptor, which again is a class of pattern recognition receptors, binds to its ligand. It will always elicit a response in that cell that will induce genetic changes to make new proteins that are responsible for eliciting a host immune response. And so one of the classic responses is the process of phagocytosis. So let's look at that process next here.